Well, welcome back. We're now in this very special session called the Visionary Keynote. And we have an outstanding, I would say exemplary speaker by the name of Professor Robert Langer. Before I get started introducing him, I just want to tell you about a few logistics. So Dr. Langer will speak after I introduce him, and then follow that by a Q&A session. And I will ask him some questions, but I encourage all of you to ask questions. And the way you can do this is through your chat. You can type in your questions, and I encourage as many as possible. And my staff and I will read those questions to Dr. Langer. But let's make it really interactive. Let's make it really natural. And let's make it very exciting. So without further ado, let me therefore introduce Dr. Robert Langer. He's amazing. Get ready for quite an amazing treat. He's one of the 12 Institute professors at Massachusetts Institute of Technology, or MIT. Being Institute professor is the highest honor that can be awarded to a faculty member at MIT. He's written over 1,500 articles, which have been cited over 350,000 times. His H index of 292 is the highest of any engineer in history and tied for the fourth highest of any individual in any field. You impressed? Well, let's hear more. His patents have been licensed or sub-licensed to cover 400 companies. And he's a co-founder of a number of companies, including Moderna, the one that has the mRNA platform that's making important vaccines. Dr. Langer served as chairman of the FDA Science Board, its highest advisory board from 1999 to 2002. He has over 220 awards, include both the United States National Medal of Science and the United States National Medal of Technology and Innovation. He's one of the 3 living individuals that receive both of these honors. He also received the Charles Stark Draper Prize, often called the Engineering Nobel Prize, the Queen Elizabeth Prize for Engineering, the Albany Medical Center Prize, Breakthrough Prize in Life Science, Kyoto Prize, World Prize in Chemistry, Millennium Prize in Life Sciences, Millennium Technology Prize, Priestley Medal, the highest award of American Chemical Society, the Gardner Prize, and Dreyfus Prize in Chemical Sciences, just to name a few. And believe me, any one of these, to me, is so much coveted. It's like having a Nobel Prize. He holds 36 honorary doctorates and was elected to National Academy of Medicine, National Academy of Engineering, National Academy of Sciences, and National Academy of Inventors, among many others. Now, you can see why we're so excited, why I'm so excited to get him to speak, because of his breadth and depth in science. As you heard, he's made tremendous important discovery work, but also went all the way to form companies and actually translate them into commercial products. So let's listen to Bob Langer. Let's welcome him. He's going to give a keynote for about 20, 25 minutes. Then I think we're going to be very excited about talking to him in the Q&A session. So please forward his presentation.
Thank you very much. Thank you very much. It's an honor to be able to present this talk to you. So I'd like to tell you a little bit about my career and what we've tried to do in our laboratory. So I'm actually a chemical engineer. I got my doctorate at MIT in 1974. And when I got it, pretty much that year, all my friends decided that they wanted to work in the oil industry. That's what chemical engineers did at the time. So I thought that's what I'd do too. But when I went to those different companies, I wasn't really excited about what they were doing. You know, they were talking about increasing the yield of certain petrochemicals by like 0.01%. And I just hoped that there would be something I could do with my life where I might be able to make more of an impact. So I applied for teaching jobs, because I was very interested in education, but nobody would hire me. And then I decided, well, maybe another way I could help people would be through medicine. So I wrote to a lot of hospitals and medical schools, and there, again, nobody would offer me a job. But then one day I remember walking into the lab that I was in, and one of the older people said to me, he said, Bob, he said, there's a surgeon in Boston named Judah Folkman, and sometimes he hires unusual people. He thought very highly of Dr. Folkman. I don't know what he thought about me. But anyhow, if we go to the first slide, Dr. Folkman had a very interesting idea. And his idea was that if you could stop blood vessels, maybe that would be a new way to stop cancer. And this is shown, this is from the New York Times, actually, many, many years ago, just showing that if you go to the lower left, a tumor cell might become abnormal. It grows in a three-dimensional mass. As you look at the arrows going to the middle, the lower center of the screen. And when the tumor gets to be about 1 million cells or 1 millimeter cubed, it runs into a nutrition problem. It can't get nutrients or get rid of waste. But what he said is that the way the tumor solves that problem is it sends a signal, which he called tumor angiogenesis factor, to the surrounding blood vessels. And normally they don't do anything, but in the presence of the tumor, they multiply rapidly and grow right towards it. And that solves the nutrition problem for the tumor. Now it can get nutrients through the blood vessels. And as you go to the upper right-hand part of the screen, it's vascularized. It gets bigger and bigger and bigger. And it can also, as you go to the top right, spread through those blood vessels. That's a process called metastasis, which of course can kill people. And Dr. Folkman's idea was that maybe if you could stop those blood vessels, achieve what he calls anti-angiogenesis, maybe that would be a new way to treat cancer and even perhaps other diseases. Now, it turns out that everybody at that time ridiculed him. They said, this idea doesn't make any sense. Of course, I knew nothing about biology. In fact, I would end up being the only engineer in the entire Boston Children's Hospital at that time. I thought it sounded fascinating. So I went to work there and we began isolating certain types of molecules. But one of the key problems in solving this problem was to develop a bioassay. And there wasn't one for studying blood vessels. So we had to invent one. And what we did is shown on the next slide. What we did is we took this was published in Science in 1976. We took tumors, V2 carcinomas, put them in the eyes of a rabbit. And what that did is normally there are no blood vessels in the rabbit cornea. But when you put this tumor in, over about a 9 to 12 week period, blood vessels will grow, as I'll show you later, from the edge of the cornea to the tumor. And so you could take an ophthalmic microscope and actually semi-quantitate what happens in terms of the length of the longest blood vessel. But the big problem we had is that you also need to have a delivery system for the molecules we were isolating, which were large molecules. And like I say, there wasn't one to do that. And what we thought is maybe we could put a little vehicle like a polymer that could slowly deliver these substances over this period. Now, when we first proposed this, Dr. Folkman was on the board of the only company in the world that had done any type of drug delivery work, and all of their work was on small molecules. And they had Nobel Prize winners and people like that on their board. But when we asked them, could they help us? They said, what's on the next slide? They said basically that you can't do this because large molecules can't slowly diffuse through solid materials. It's kind of like any of us walking through a brick wall. We're just too big. And the literature actually said something similar. Next slide. That the use of these systems for slow release has been virtually restricted to small molecules. Sometimes I think the only reason we maybe succeeded is I hadn't read any of this. So I tried anyhow. I spent about 2 years working in the laboratory with different materials, different ways of doing this. And I actually found over 200 different ways to get this to not work. But eventually I was able to make little microparticles or nanoparticles. Next slide. And here you see a picture of one on the left and it cut in half on the right. And in Nature in 1976, we published the first paper showing that you could take these tiny particles, next slide, and release them for really any period of time. Also in that same paper, we showed that you could release for the first time nucleic acids like DNA or RNA as well. And later on, we even figured out ways to control kinetics, like at constant release. The next slide shows an example of that. Here's a constant release of albumin, 68,000 molecular weight. So we had done this work, but one of the things, of course, you do when you're a scientist is you present this work at different meetings, and so we published this in Nature. I remember in 1976, I gave a talk on this in a big material science meeting in Midland, Michigan, and I was 27 years old, and I was very nervous, and I got up and I gave this talk, and I thought I did okay at the end. I didn't forget too much what I was going to say. So I thought when I was done with this talk, all these older scientists, being nice people, would encourage me, this young guy. But when I got done, I stepped off the podium and a whole bunch of them came up to me and they said, "We don't believe anything you just said." On the next slide, this is what they said. Again, they repeated what I mentioned before, that the large molecules couldn't diffuse through solid materials. They also said that we were using organic solvents, and that would denature whatever we put in, peptides, proteins, or nucleic acids. And actually, things even went downhill from there. I really liked being a postdoc, but a lot of my friends said I shouldn't do that forever, so I started applying for faculty jobs. But no chemical engineering department, which was my discipline, would hire me. I also wrote research grants, and my first 9 were very badly rejected. They said chemical engineers can't do this kind of bio stuff. And in terms of a job, I finally did get a job in the nutrition department as a young faculty member. I think the reason I got it was because the head of the department liked me, and Nevin Scrimshaw, but he was kind of like a maybe what you call a benevolent dictator kind of department head. Since he wanted to hire me, he offered me a job, but he didn't bother to ask the rest of the department what they thought, which may have been okay, except for one reason, which is the fact that the year after that I joined the department, he left. So a lot of the senior faculty decided to give me advice, and their advice is I should leave too. In fact, the next slide comes from Michael Marletta, a good friend of mine, also a member of National Academy of Medicine and National Academy of Sciences, but he was talking about in 2014 when I received an award, and he was reminiscing about what it was like to be a faculty member with me. And I'll just read this to you. This is a direct quote. He was talking about what it was like to be with me in 1980. And he said one evening he went to a faculty dinner at a Chinese restaurant with me and some senior MIT professors. He said a senior scientist sat quizzing us while smoking a cigar. He said when the older scientist heard my concepts for drug delivery, he blew a cloud of smoke in my face and said, "You better start looking for another job." So this was all, not very positive, but I, sort of put my head down and kept on going. And of course, we had done, and I believed in the work we were doing, and we had shown that you could use materials to deliver molecules of any size. And so we were able to develop the bioassay that I mentioned, which is shown on the next slide. And again, this is from that science paper. But now I'd also isolated all these fractions. And I just want to, again, show you one picture. If you're squeamish and don't like the sight of blood, don't look, but there's no other way to illustrate stopping blood vessels. So this is from that same paper in Science in 1976. We actually tested 2,000 rabbit eyes. And on the left, what you see is one example. We did almost so many controls and so many fractions didn't work. And this is at 9 weeks out. And if you look at the left-hand side, you see a white part in the eye, which is the tumor, and you see the sheet of blood vessels growing right towards it. And if you looked at an eye like this just 3 weeks later, it would be the tumor would be out of the orbit of the eye. It would be three-dimensional. It would actually kill. We sacrificed them before that. But what was so striking is when we found one of the fractions, like I say, 99 or so didn't work, but one of them did. And what we were able to show, and we are able to do this even quantitatively by measuring the length of the longest vessel, we were able to show that there is R inhibitors for the first time that and have bioassays for them. And what you see is in the right-hand picture, the vessels are lower, they're sparser, they avoid the tumor, and 60% of the time these tumors never got big, whereas 100% of the time, 1,000 out of 1,000, the controls always got big. So this showed for the first time that these molecules did exist. It still took another 28 years and wonderful work by Genentech and other companies using some of the technologies that we had developed to actually get an FDA approval for an angiogenesis inhibitor. But starting in 2004, 28 years after this paper, it was striking what would happen. Next slide. And what you see is starting in 2004, one after the next, blood vessel inhibitor approved by FDA. Avastin is like the number 2 or 3 biggest selling, most widely used biotech drug in history. And it's not just that, there are many others that have been approved by the FDA for all kinds of cancer, and not only for cancers, but for eye diseases like macular degeneration and diabetic retinopathy, some of the most common causes of blindness in the world. There are drugs like Lucentis and Eylea that are blockbusters, widely used, and actually the first substances that could stop blood vessels in the back of the eye, and in some cases even reverse blindness. So that actually was very exciting, even though it took a long time. Now, Dr. Folkman and I also thought, we should file a patent, which might seem common today, but in the 1970s, Boston Children's Hospital, which is a great hospital, never had a patent. So we filed a patent for the first time. And 5 times in a row, the examiner rejected it. And everybody told me, again, I should give up because I was wasting so much money. But I don't like to give up. And so I started to think, how could we convince the patent examiner to allow this patent, legally, of course, and just explaining this by science really wasn't working. So I, as I told you in the beginning, when we first started doing this, everybody told me this was impossible. It could never work. I wondered if anybody ever wrote that down. So I did what's called a science citation search. Now in 1981, 82, 5-6 years after we published the papers in Science and Nature, to see if anybody had written anything. And on the next slide, I want to show you what we found. And I had no idea this was written, but it was from 5 different polymer or material sciences talking about the drug delivery work we'd done. And they basically are saying, generally the agent to be released is a relatively small molecule with a molecular weight no larger than a few hundred. One would not expect that macromolecules, for example, proteins, could be released by such a technique because of their extremely small permeation rates. However, Folkman and myself have reported some surprising is a really good word for a patent examiner, that clearly demonstrate the opposite. So I showed this to the people in the hospital, and they showed it to the patent examiner. He said, gee, he had no idea, but he said he will allow this patent if I can get written affidavits from each of these 5 people that they really wrote that. So I wrote them all. I was this young professor. I wrote them all, and they were all nice enough to write me back that they really wrote this. And we got this very broad patent, next slide, on first time for delivery of large molecules like proteins or DNA or RNA. And with that, we actually licensed it to large companies, but they didn't use it that much. And I really wanted to see our technology get used. So I got involved in actually helping start companies myself. And one of them, next slide, and I've almost always done it with my students, but one of them, it was Enzotek, which later merged to become Alkermes, and they have, now 25 products that are FDA approved or in clinical trials. It's led to very widely used new treatments for schizophrenia, alcoholism, opioid addiction, diabetes. They have about 2,000 employees. Some of the products they've developed are on the next slide. And other companies too have used these principles, and we had arrangements with different companies to help them develop these things. But it's led to, just a few examples. On the bottom left is a Risperdal, which is a multi-week treatment that you inject little microspheres for treating schizophrenia. In the middle of the screen is Lupron Depot, which we helped Takeda with. That was originally a once-a-month treatment, now once every 6 months injection for treating prostate cancer, endometriosis. The work also led to drug-eluting stents and many other things. But even having done this, we all, and even having made nanoparticles back in the 1970s, sometimes when you inject them, they didn't last that long. So we developed another way, because macrophages would eat them. So we developed another way by coating them with molecules like polyethylene glycol to make them last longer. And I just thought I'd show you a video that Nova, the TV show, did on some of our work. Next slide, our next video.
He starts with a nanoparticle of anti-cancer drug. That gets encased in a plastic that releases the drug over time. That in turn gets a special wrapping that disguises the package as a water molecule to fool the body's immune system. And last but not least, the address where it should be delivered, a key that will only fit the lock of cancer cells.
I should point out, it really doesn't blow the cell up that way. At least that's what a lot of the medical doctors I work with have told me. But you can use this approach, creating nanoparticles with polyethylene glycol on them, whether you make them out of polymers or lipids, to deliver small molecules. But what's really exciting, and what I want to move to next, is that you could even use them for genetic therapies, like delivering siRNA or messenger RNA. And in the first case, siRNA, there's a company, Alnylam, that I had the good fortune of being an advisor to. And by the way, I should make a disclosure. I've had I've been paid by all these companies that I'm mentioning, Alkermes and Alnylam and soon Moderna, which I'll mention as well. But basically, what you have is you could use this for genetic therapy. It's absolutely critical to have them in nanoparticles, otherwise they'll get destroyed. And Alnylam, where one of my students led this project, Akina King, they were trying to treat a rare disease, ATTR amyloidosis. And so they put the siRNA in the nanoparticles, in this case, lipid nanoparticles, and that's led to a product that's helped many, many people. Next slide. It's called Onpattro. It got approved in 2018. And that began to show that you could start to use nanoparticles to not only deliver, or microparticles to not only deliver small molecules or proteins, but even, in this case, genetic materials. Now, in 2010, going back a little bit in time, Derek Rossi, a young scientist, came to see me for advice about possibly starting a company in the area of messenger RNA. And if we just go to the next slide, what you see is the whole idea is that DNA, I'm sure you all know this, RNA makes protein. And of course, protein, many companies, Genentech, lots of others, Amgen, have come up with all kinds of protein therapies. Most of the most common and widely used drugs today are proteins, like different antibodies. But one of the things that Derek and I began talking about, along with Nubar Fayan and Ken Chen, is, could messenger RNA be a drug? And the next slide shows that that basically, if it could be a drug, not only would you be able to maybe make it really fast, because you could make drugs really fast, because see, proteins might take a year to make as you grow them up, and whether it's through eggs or fermenters or whatever, but and purify them and so forth. But if you could make RNA, all you'd have to do is inject it into the body, protected by a nanoparticle, and the body would be the factory. It would make the drugs. So the idea is that you could hopefully make new drugs or vaccines very, very quickly, really in weeks, and it could enable new kinds of protein therapies. It would also enable you to treat diseases that could never be treated before, like intracellular and membrane-bound proteins could be used to treat diseases. So in 2010, the four of us who I mentioned started a company called Moderna. And I should point out that for a long time, Moderna was widely criticized. All the stock analysts and many newspaper reporters would keep saying that this kind of technology will never work. In fact, even a year ago, when we started doing the work to create the vaccine, the same thing happened. I'll just show you a newspaper article on the front page of the Boston Globe, my home newspaper. Next slide. And this is the front page headlines with my picture on the front page saying, this, note the date, May 19th, 2020. And all these people are saying, this is not how you do science. So that was very discouraging. But the terrific people at Moderna, headed by Stéphane Bancel and Stephen Hoag and others, never gave up. And as on the next slide, what you see is a picture of the vaccine that's been approved for emergency use authorization and been used all over the world. I should point out the other one, that BioNTech and Pfizer, that also uses nanoparticles to deliver it. So it's been wonderful to see this actually happen. I thought I'd just conclude really quickly by mentioning one other area that we've worked in, which is tissue engineering. Here I've worked very closely with Jay Vacanti, also a member of the National Academy of Medicine. He's a surgeon, and one of his patients is shown on the next slide. He's a little baby dying of liver failure. And Jay once asked me, could we ever make new tissues and organs for people like this? And on the next slide, from a paper he and I wrote in Science, this has been cited over 8,000 times, we had the idea that you could take any cell type, but if you take these cells and inject them at random, nothing happens. But these cells are smart. So if you put them close enough together, say on a scaffold, they could actually form tissue structures. And then you could grow them in a bioreactor, say in vitro, and then make virtually any new tissue. By the way, when we first proposed this many, many years ago, there were, again, enormous skepticism. But just to show you a few pictures, the next slide is a picture of some of these fibers with liver cells on them. And I thought I'd just give you two examples of it. In one case, and this is now FDA approved, in one case, you could make new skin for burn victims or people with skin ulcer, diabetic skin ulcers. The next slide shows a picture of a little baby who's very badly burned. But you could take the product, next slide, which is neonatal fibroblasts on this scaffold, put it on the child in the beginning, next slide. And if we come 3 weeks later, next slide, he's doing better. And 6 months later, on the next slide, he's pretty much healed. And you can use these. There's various trials going on for making blood vessels, again, by many of our students, many people who are at NAM, to make blood vessels, spinal cord repair, new pancreases, all kinds of things to help restore hearing, to help patients using tissue engineering. Also, there's the possibility, and I'll end on this, of maybe even someday making organs or tissues on a chip to greatly accelerate drug testing and maybe even reduce the amount of animal testing and speed up human testing. So again, a few other of my students, Gardana Vanyak, who's also in NAM, she and some of our colleagues actually made a heart on an in vitro heart. And the next slide shows a picture of that. You have the sarcomeres and intercalated discs, but I want to show you a video of this heart on the next video slide. And here, we're not shaking this or anything, but there's 6 million cells here. And see how they're beating all as one. And so you can literally have do this in vitro. And you can take that, and on the final slide, what I'm going to show you is that you can take an EKG of this. And it's like a regular EKG. And in fact, you can do drug testing on this. And many companies are using this type of thing for drug testing. There's also other companies that are making, and scientists who are making livers on a chip, making a gastrointestinal tract on a chip. We're even working with Lilo Esai at MIT to try to make a brain on a chip. So there are many, many exciting things that I hope are coming out of this. So I'd like to conclude this talk and just thank you so much for listening to me. You know, it's really been an honor and a pleasure to go over the work we're doing. And I wish you all the best of luck at this meeting. Thank you so much. Wish you all the best of luck at this meeting. Thank you so much.
Bob, thank you. It's unbelievable, so inspiring. I gather that you're having trouble with video, so let's just talk by audio. You know, you bring back a lot of memories in your talk for me. Judith Folkman. I was working in Cliff Barger's lab, you remember? And
I do, Victor, yes.
Yes. And by the way, that Boston Globe picture of you about Moderna, the background is Cape Cod. I can recognize it.
That's exactly right. That's my house in Cape Cod. Very, very good.
Yeah. Okay.
Well, you're terrific. That's exactly right.
So I would say that I was reading your introduction and blown away by the fact that you have license or sub-license to over 400 companies of your invention, aside from, of course, your index and the many, many awards that you have founded. I think we asked you to give this visionary talk, and you did precisely that. That is to give people an idea how actually, when you are committed to science, when you have an idea committed to, sometimes it doesn't always get the kind of recognition or support, but then you were able to do things differently from many others. I think the other thing I learned, of course, is the interdisciplinary nature of your work. Engineering, biology, medicine, and when they all come together, it's just so beautiful. So I'm going to ask you a few questions, and then there will be possibly audience who would ask you questions. We use a chat function, so we'll see.
Okay, that sounds fine, Victor.
Yeah. So my question for you is maybe speaking to the young innovators, as you so graciously agreed to do, how do you decide what question to ask in your research, what to investigate? In other words, did you have a very clear idea of the steps you want to go forward, starting with a scientific question, or in fact, you have this great foundation, but look for bold and new ideas and opportunities?
Yeah. Well, I mean, that's a great question, and there's not a simple answer. I basically try to think in my own mind what will have the biggest possible impact, either from a basic science standpoint or ultimately applied clinical standpoint. But it's I guess an educated guess on impact.
Yeah. So, for example, what in the very beginning of your career, what made you decide to say, yeah, I'm going to work on clinical medicine, as you said, and secondly, I work on these things nobody's done before on angiogenesis, on the release. How do you get into that kind of thinking?
Sure. Well, for me, actually, that's an interesting story. When I was a graduate student, I'm a chemical engineer, and all my friends would go into the oil industry. This was the early 1970s, and I really wasn't excited about that. I was, again, looking for something where I felt I could, do some more good for people, to have some impact, which could have been education, could have been medicine, but it wasn't. What it wasn't was like increasing the yield of some petrochemical by 0.1%, which a lot of those jobs were, and they were very high-paying jobs. But I was really looking for something where I guess I felt I could make a difference in people's lives. And actually, I'd like to tell you that a lot of that, I got 20 jobs for oil companies. Nobody in education or medicine would hire me. And Judah Folkman was a visionary guy, and sometimes he would hire unusual people, as people told me. And he was nice enough to hire me, and I thought his ideas were just terrific, and that's why I ended up doing that.
Yeah, so my question is, how do you balance between kind of basic research, curiosity-driven research, which you clearly do, to think about it has to be translated, and how do you actually interface, if you will, from the academic to the commercial world?
Yeah, well, so basically the way I look at it, at MIT, my academic lab, we're doing basic research in either engineering or medicine, more fundamental research. But I really don't want to just leave it there. I really want, again, to have our work make a difference in people's lives. And I realized early on, I thought if I just published, everybody would use things. But that's not true for the most part. I mean, maybe for 1% of people's discoveries that happens. But for most things, I found if you're not your own champion, nobody else will be. So I started in the 80s, patenting things. And then I thought even large companies would use them, but then they give up very easily. So I got involved working with my students to start small companies, and those companies, some of them did quite well. But it was really because of these patents and because of the small companies that I was able to do more of the translational work.
Yeah, I mean, I think to me, there is a lesson that people can learn, right? That established institutions are not quite willing to take risk. And when they look at success, they define it in a very, shall we say, traditional way. So whereas new ideas that come up requires this kind of stepwise, but let's take alchemies. Let's take Alnylam, which, by the way, I was on the board.
I know.
I joined this. And then let's take Moderna. What happens is that when you begin to invest in smaller, more people believe in technology, being able to take it further, it can become as big as you can imagine. But it takes a more innovative approach. And what you're seeing, Bob, and this group of people who you're speaking to, I guess a lot of them are like you, because they're willing to take this $50,000 award from us, not a lot of money, just to get started, right? And you see the range of ideas is quite amazing, some of which actually overlaps what you talked about, and just to get started. And I always say that's hardly enough money if you were to put a traditional grant to NIH or whatever. I mean, how do you do biomedical research with that kind of dollars? But it gets people going. And probably that's how you start, right? An idea. Because when I was at Duke, we would give out $50,000 seed money, and you'd be surprised how many young people apply for them. Now, so I think there's a lot of lessons here. How do you take a nascent idea? How do you get some money to test it? And how do you actually develop it further, as you have done, into much larger scale application? So tell me, what are you excited about right now? What's next for Bob Langer?
Well, we're doing a number of things. I mean, in the lab, I'd say there's 2 or 3 big things. One of the things we're doing is a lot of work with the Gates Foundation on developing, new medicines, new vaccines, new ways of giving vaccines, and nutrition for the developing world. And a lot of it's been, kind of basic things, but companies have spawned out of that as well. And so now we're like, for one example is that when we published some of this in Science a couple of years ago is that we could give a vaccine with a single injection, but it could have like 12 pulses, and so it's a self-boosting vaccine, and that could improve patient compliance and also could get higher titers. And so that's just one of a number of examples that we're working on with Bill and his team. And there's also new ways of giving, nutrition to combat malnutrition. And we've published these things like in Science or Science Translational Medicine or Nature. But we've also started some companies on those as well.
Oh, so listen, there are questions coming in. So I'm going to ask Michelle Topless, the director of the program, to ask some of the questions. They're very eager to ask you some of what they can learn from you. But let me just say one thing about Gates Foundation. Yeah, I'm very excited, for example, the whole idea of in vivo gene therapy they're working on, being able to take gene therapy and being able to take gene editing to treat sickle cell anemia, which, as you can imagine, thalassemia, which are prevalent in Africa. Most people argue that technology that you have developed are too expensive, right? And how do you actually give it to the rest of the world? But I think you've proven that it can be done. Can you say a word about that before I turn over to questions?
Absolutely. So we've developed new materials, inexpensive materials, more inexpensive manufacturing procedures. And like I say, even ways like I'm mentioning with the vaccines, where somebody wouldn't have to go back to the doctor. And so those kinds of things are, I think will hopefully also make it less expensive. Also new things like microneedle patches that, might be much easier and cheaper for patients to use.
Beautiful. Okay, Michelle, there are questions coming in from the audience. Go ahead, Michelle.
Okay, great. Thanks so much, Dr. Zhao. 1 minute. So, Dr. Langer, thanks for answering these questions. I'm going to start reading some of the ones that have come in. Can you comment on the use of biomimicking nanoparticles, which are coated with blood and cancer cell components? And do you think this is a better approach than totally artificial nanoparticles in terms of targeting to the treatment sites versus intravenous injection?
Well, I think it's a very interesting idea, and I think it's hard to say whether it's better or not. I think it's at an earlier stage, but I think it's a good idea and well worth exploring.
Next question.
Yep. So in one of your first few slides, you showed a range of nanomedicine-based drugs used for different diseases. One would have envisioned high efficacy of these drugs in tackling these different disorders, yet we don't see the disorders or diseases being eradicated. So is there a missing link somewhere, or is the gap from bench to bedside still too large?
Well, I think it is too large, but of course, and I agree that drug therapies are not a cure, but vaccines possibly can be a cure. So that's another thing that obviously the nanoparticles can be very useful for. That may not be a total cure, but it can go a long, long distance towards it.
Great. Another question is, advances in biomedicine and public health have prevented or alleviated much suffering, but perhaps indirectly increased many of the ripple effects of longevity and negative human impacts on the globe. Are there any promising approaches to modulating rather than maximizing human populations to hopefully avert our extinction?
Boy, that's a tough question. I'll give that one to Victor. He'll do much better than me.
All I want is clone you, Bob.
And vice versa.
How about this? You were very fortunate to have your work accepted for publication in Science, Nature, etc. And this would have greatly increased your credibility and enhanced your pathway up the academic ladder. What about many of us who do not have the privilege of publishing in these top-tier journals? Can you provide any guidance?
Well, first, I think publishing, period, is good. But actually, I should tell you, when I started, I got rejected many, many times. And I just kept trying, and I kept learning from what the reviewers wrote and the editors wrote. So I don't think just because I mean, those journals will reject everybody, and that's okay. It's not, to me, it's not how many times you get rejected, it's how many times you get accepted, and that's true in many things in life.
Hey, Bob, why don't we do this? Why don't we just, because time is running short, why don't I give you the last word? What would you like to say to the innovators? Give them some words of wisdom.
Well, sure. Well, I mean, I guess what I would just say, and this is something I've said in some commencement speeches, and I believe in this for almost anything you do, is it's really important, I think, to dream big dreams that can change the world. But you should recognize that if you try to do that, you'll probably run into a lot of opposition. People will tell you your ideas are impossible. And don't give up. I mean, keep trying. Even that last question relates to that. You're going to run into all kinds of roadblocks and failures, but the goal is to win the war. Not, even if you get setbacks, just keep trying and do something important that'll make the world a better place.
That is a great way to end. I would say the lessons I learned from you, by the way, even though we're almost contemporaries, I see you as a role model. I mean, you've done so much in bringing the sciences together. But I think the main thing, if you said repeatedly, is trying to make a difference to humankind. I think that purpose in life is so important. And what you do in research, in this case, we talk about healthy longevity. People are finding ways to say, can my good idea improve people's quality of life? And even as they live older, can they become healthier? I think that as long as people have that kind of real focus and value, and then bring your science to it and understanding that you can't solve the problem alone, we work with others, I think you're the exemplar of all these things. So thank you, Bob, so much. I know you're such a busy man. Just to get the time from you is greatly appreciated. And I'll say that personally, you're a mensch.
Well, it's an honor, Victor. You're a wonderful person and a great leader. So it's my great honor to be able to speak to you and everyone else. Thank you so much.
Thank you very much indeed. And take good care.
You too. Bye-bye. Thank you.